stop my video for the presentation. Awesome. So thank you so much for introducing me and for inviting me here. So my name is Heather Ward, and I'm going to tell you about developing scalable standards-based workflows using workflow description language in Docker. And workflow description language is just a particular language used for writing pipelines or workflows, and you might hear me refer to it throughout the talk as WDL or WDL. So I will try as much as I can to say the full name, but if I say WDL, that is what I am referring to as the workflow description language. So this is the kind of the structure of my talk. First, I'll give you a brief background of my career progression here and what I do in my day-to-day. And then I'll talk a little bit about standards in open science and why they're important. And then I will deep dive into a particular language, the one that I use every day, which is workflow description language. And then I'll talk a little bit about containerization and why it's important. So I'm the bioinformatics lead currently at DNAstack. You heard already a little intro of my path through there. For many, many years, I worked completely alone as the only bioinformatician here. And DNAstack is largely a software company. So their mission is to accelerate scientific discovery by connecting the world's data. And the way that they do that is they try to build software that enables the sharing and discovery of data, largely in the genomic space. So the vast majority of the team are software developers. And then there are a few scientists in there that our goal is really to both show how the software works and how it can enable discovery and help kind of onboard different labs or researchers into using it. And also, when we're not doing other things, we'll run sort of internal analyses and our own sort of research projects. So for example, during the COVID pandemic, we really quickly pivoted into just sequencing and assigning lineage and calling variants on as many COVID sequences as possible and sharing those back to the community. So just again, to show kind of what our software is able to do and to enable that sharing to the research community at large. So a day in my life at DNAstack usually looks like mostly writing workflows. So in some cases, that can mean that a collaborator that comes to us, so a university lab or a hospital, for example, might have some sort of analysis that they already have in mind. And what they want to do is scale that up, put it in a cloud-ready format so that it's both easier to share with collaborators and can run maybe more parallel. So in those cases where they already have an analysis in mind, I will take their local running pipelines and I'll just convert them into workflow description language-based workflows. I will test them in the cloud, make sure that they run reproducibly and validate them, and then I'll hand that back off to them. In other cases, though, our collaborators have perhaps some sort of raw data and they don't maybe know exactly what sort of analysis they want to run. They just have an idea of what the outputs might look like. So in those cases, I'll research maybe the best practices pipelines that might exist for that type of data, tools and analyses that they might want to use to write those into workflows. And I'll kind of present that back to our collaborator and see what they think. And when we get the go-ahead, I'll then write that analysis into a workflow that runs in the cloud. I also spend a good amount of time running workflows. So in some cases, our collaborators, they want us to containerize their tools. They want us to write their workflow for them, and then they will themselves run it. But in a lot of cases, they want to be completely hands-off and all they want is the output data. So in those cases, I will both write and validate the workflows for them, and I'll also run them at scale. I also spend a good amount of time optimizing workflows. So this can be for a number of things, reducing cost, reducing runtime, while also maintaining accuracy of the workflows. Also, as I mentioned, since bioinformatics, as you know, is a very, very broad topic that covers a lot of different data types, disease areas, that kind of thing, I am not usually the expert in the room for any number of these different data sets. So we take on a lot of different projects with a very, very varied kind of set of data that we're looking at. So a lot of my time is spent doing literature reviews. I also spend as much time as I can trying to figure out the best tool for different tasks. So for example, if we're looking at variant callers, trying to compare and contrast the current best standard variant callers that there are. I look for new tools that have been published recently, updated releases to see if I can keep on top of that. I also, as I was mentioning, I really try to keep up to date with pipeline optimizations, and that might be something as simple as swapping out a library or dependency to make things run faster, but it might be a greater change like updating a tool version or just choosing a different tool for a different step. So that's largely what I do. Again, there's a lot of different collaborators that we have, and it's always kind of a different area of disease, different genetic structures. As I mentioned recently, I had to really quickly take a crash course in viral genetics because going into COVID, I definitely did not know very much about viral genetics at all, other than that they had a genome. So it's kind of different projects all the time, which is really fun for me at least. All right, so standards in open science. So I've got a quote here from Michael Nielsen, who says that open science is the idea that scientific knowledge of all kinds should be openly shared as early as is practical in the discovery process. So the key point there is as early as is practical, and that's really important because there's a lot of data out there, but unless it's shared back to the community in a timely fashion, there's going to be a lot of repetitive and maybe unnecessary research that is going to be done. There could be discoveries that could be held back for years at a time, and in some cases, they can be life-saving discoveries. So for example, we can consider the response during the early days of SARS-CoV-2. This is a case where scientists and governments around the world were able to really rapidly open up their data. And because of that speed of sharing, we were able to kind of track the migration and the evolution of the virus almost in real time. So sharing often and early was really beneficial for everyone. And with open science, you get greater efficiency and faster development because, for one thing, you don't need to reinvent the wheel. As I mentioned, you don't have to repeat experiments that may have already been done and concluded behind closed doors. We also know that technology and discoveries build one on another. So if discoveries are open and freely shared, researchers can be allowed to iterate much more quickly and move much faster than if they're only able to look at their own work in a sort of vacuum. So in addition to greater efficiency and faster development, open science also allows for greater scrutiny and accountability. And those are both really good things. For example, if we consider encryption algorithms, the paradigm with modern encryption algorithms is that the algorithms themselves are published openly. So how they're enacted is published. And the public understands exactly how they're implemented. So the idea there is that you want to hide the key but expose the algorithm. And you might ask, why is that a good thing? And the answer is that if you're developing a new algorithm for encryption and you and maybe everyone on your team are trying to break it in every way you know how, all that you're really proving there is that no one in the room knows how to break your algorithm. Whereas if the algorithm is made public, you know that there's going to be many, many people around the world with perhaps nefarious intentions that are going to be trying as hard as they can to, and failing to break it. So it's that reality of people around the world, the entire community really trying to break it, that is giving you confidence that it's actually secure. And finally, open source and open science have been proven to be profitable. When everyone has that open mindset, everything happens faster and more effectively, and it genuinely is better for everyone involved, and that's been shown time and again. So these are some examples of global collaborators that are working to make their data accessible. There are petabytes of data that are being made publicly available to the medical and scientific communities, and these represent literally billions of dollars of publicly funded research. So citizens of countries around the world are paying taxes, they're donating to charities in order to further these discoveries. And I think it's reasonable that most people would want their data to work as hard as it can for their donations. So similar to building an encryption algorithm and only having your teammates try to break it, if you generate an incredibly valuable data set using thousands or millions of dollars of public funding, then you only let the people in your own lab access that data to try to glean insights. That's probably not the most effective way to get the most out of that data. And I do also understand that there are conflicting interests between generating and sharing data openly. And a lot of these conflicting interests are kind of inherent in the way that both academia and industry are set up. So for example, people don't want to be scooped, and that might be publishing a paper on their data or developing a new molecule or anything like that. But it can't be denied that removing some of those boundaries to data access is inevitably going to increase the speed of research and discovery for everyone. So the less boundaries, the better. Here, I just want to look quickly at a particular case study and focus in on the MISSING project. So MISSING is a huge collaboration between scientists, hospitals, sequencing centers, charities, and also industry partners that seeks to explore the genetic causes underlying autism spectrum disorder. And you might be aware that ASD is a really complex genetic disorder. And despite it being clearly heritable, the mutations known to influence ASD explain only a tiny, tiny fraction of ASD cases. So because it's so variable and expresses itself in so many different ways, it's really important to have a very large quantity of both high-quality genotypic and phenotypic data. So MISSING's ongoing goal is to gather as many trio datasets as they can, that being an affected child and their parents, so that they can explore this known heritability of ASD, so that they can look for de novo mutations, maybe structural variants that might help to elucidate more of the genetics underlying ASD. So a few years ago, the MISSING collaboration came out with over 10,000 whole genome sequences for these trios in a database they termed DB6. It's a really groundbreaking data set. It can be freely explored by ASD researchers. But even with that many trios, we understand that it's really just a drop in the bucket statistically when we're looking at such a complex disorder with so many potentially small additive effects across the genome. So in order to really drill down on some of the genetics and to cluster these individuals into different hierarchies, we'll really need tens of thousands more samples. But even aside from that, there's something else to consider, and that's that this data set is made up of individuals affected by ASD and their parents. And typically researchers who want to understand ASD are the ones looking for access. But it's not only useful to ASD researchers. There are 10,000 high-quality genomes with associated phenotypic data. So it might be interesting, for example, to researchers studying neurodivergence or behavioral disorders in general. It might be interesting to researchers studying population genetics, or those who want to study structural variants or inheritance, or perhaps even researchers who want to use the data set as a control set when they're exploring another group with a different set of phenotypes. Because, of course, we also have the unaffected parents in here. So the point is that there's a huge amount of time and effort and also money that has been devoted to generating this data set. And if only ASD researchers were able to find it and to search it, I think there's a lot of potential value that's being left on the table. Oh, sorry, excuse me. So why am I bringing this up? Well, there's this idea of data silos, and that is data that has been produced and that may have value across a wide range of applications and to a variety of researchers, but that for one reason or another, it's isolated and cut off from the greater pool of research data. So it might be siloed because, as with the ASD example, maybe it's been produced for a certain purpose and only people studying that area may be aware of it. It might also contain protected health information, which means that sharing it is much more difficult. In some cases, also data produced in a certain country may not be allowed to physically leave the boundaries of that country. So that can make it really difficult for international collaborators to kind of access those insights or to, for example, co-analyze that data set with their own. It also might just be sitting on a hard drive in a lab, awaiting a paper to be published, and it might actually never touch the internet. So there's a whole number of reasons why data might exist in a silo. But we know that open science and the open sharing of data as early as is practical is really the only way we're going to be able to fully take advantage of it. So having that open access and sharing is going to increase the speed of discoveries for everyone. But aside from all of the physical reasons why data may exist in a silo, there's also a much simpler reason. And that's just the fact that there is a lot of data. There's a lot of genomics data. It's very large and it is difficult to discover and to cohere. So even if every lab in the world agreed today to the open sharing of their genetic data, that doesn't really solve the problem. We would kind of be left with this huge mess of FTP servers, websites, cloud buckets, local file shares, with no real way to connect, discover, and filter and compute upon all that information. So here's the question is, assuming that researchers are willing to share their data, how do we actually make it useful and connect it? And the answer, or so a lot of people think, is standardization. So standards exist everywhere in the world. They're how the world communicates via the web and other protocols. They're how we communicate measurement information, how we exchange information across geopolitical boundaries in the web. So for example, W3C defines a number of standards for how communication on the internet should behave. You might have heard of them. If everyone decided to use a different protocol to communicate over the internet, what would happen is inevitably the web would kind of be broken up and sharded into a million fragments that weren't really able to talk to each other. So you'd have these individual disconnected groups that would, in essence, be siloed from each other, not able to share and contribute to the greater whole. And that's the state of a lot of big data in genomics today. The data sets exist. They're very large, important data sets. They, in many cases, cost billions of dollars to produce. But they're very, very large in size, and they're difficult to discover and share. So how can we take this data and make it discoverable and usable by researchers to accelerate science using data that already exists and also to enable that for data that will exist in the future? The answer is, of course, to use standards, in my mind. To do the same thing that we've done across many, many other industries. to create standards for how genomics data is communicated, discovered, and shared. And there's a ton of different standards in genomics already. The aim is just to develop more with the end goal of being complete interoperability by the individuals and organizations that choose to implement these standards. So one place you've likely already interacted with standards in genomics is file formats. There are well-defined standards for the types of files that are used to store certain types of genomic data, for example, FASTQs, BAMs, and VCFs. And when the community agrees upon a standard, this really allows the community to thrive because tools can be built that use and implement those standards as a basis to create these interoperable pipelines, analysis methods, and tools. So that's how we're able to define an analysis workflow and swap out different tools at different stages, because we have this agreement about the types of files that those different tools will operate on and produce. So we're able to kind of pick and choose which tool to use at which stage of the analysis. So that gives us a lot of power. So if you have these standardized file formats, analysis methods, etc., it means, again, you're able to really easily interoperate between all these different tools, inputs, and outputs, because they're all consistent and standardized. In addition to standards for file formats, groups in genomics are also looking to standardize things like object versioning, APIs for workflow execution and pipelining, how data is queried and discovered, and many, many more. So I just want to call out here that there's a really common impulse that many software developers and bioinformaticians will probably have at some point. Maybe you've experienced this in your own work, where you'll encounter a particular analysis and think, maybe if the data was formatted this other way instead, it would be easier to feed into my tool. So you go about defining the format of the file types you expect as input, and before you know it, you've created your own bespoke version of the VCF spec. I really urge you to fight that within yourself. In some rare cases, something novel does need to be invented, but I urge you to check that impulse within yourself and use the standards that already exist, because the whole community will be better off for it. It means your tools will be compatible with other groups, pipelines, and analyses. Your outputs can be combined with those of other labs and processes. And if there is a true need, if there is something lacking in that spec, it's worlds better if you can find the specification that you want to modify and figure out how you can make suggestions or even get involved in the decision-making process for that specification, rather than going rogue and trying to branch the spec. So since standards are used by communities, they need to be defined by them. So in the world of internet communication, as I mentioned, that standards body is W3, but in the genomics world, it's the GA4GH, the Global Alliance for Genomics and Health. So the Global Alliance for Genomics and Health is an international interdisciplinary organization. Again, they're trying to define the standards governing health and genomics data. They have over 500 member organizations, and those are pulled from academia, industry, pharmaceutical companies, individual contributors, kind of all the way across the spectrum, and in many, many countries around the world. DNAstack is involved in many of the specifications, but in particular, we lead the Discovery Driver project. So here I'll briefly discuss one of the specifications that's of particular import to bioinformaticians, and that's the Workflow Execution Service, or the WES specification. So the GA4GH gives a little synopsis of this spec. They say that the Workflow Execution Service API provides a standard way for users to submit workflow requests to workflow execution systems and to monitor their execution. So ultimately, this specification aims to define the way in which information about workflow execution, that being channeling input through various tasks, commands, and steps into some sort of outputs, is handled. And one of the key features and the goals of the WES specification is that it defines an API, or an application programming interface, that's consistent regardless of the underlying workflow language being used. So the API allows users to submit workflow runs, to monitor those runs, and to collect outputs once they're complete. And the WES spec, where implemented, allows organizations to run across different backend environments. So for example, different clouds, the HPC, regardless of where their underlying data is stored. So adhering to this spec, the WES spec, forms a sort of a contract. You know that if WES is implemented in a particular environment, the user can be assured that their workflow is going to execute there, and they don't really have to worry about the particulars of what is going to go on in the background for task submission, task scheduling, that kind of thing. All the user needs to understand is that the API exposed by the spec in order to run their analyses across any different environment that implements it. So having this standard way of executing workflows allows for a much smaller interface that the user needs to have expertise in. They only need to know how to use that API, and they know that it will work the same in Azure, Google Cloud, HPC, locally, kind of anywhere that that spec is implemented. The WES spec also integrates and interfaces with other GA4GH standards, such as TESS and DIRS. which I won't go into much detail about here, but briefly, the task execution service, or TESS, covers batch execution tasks. So this is individual sets of inputs, outputs, and containers and commands to run. It's kind of, you can think of it as a subset of WES in that WES uses it to call individual tasks. It also provides logging and other metadata. And then there's the data repository service, which allows data to be registered and consumed using consistent URLs, regardless of where it's actually stored. So for example, if a user has a DIRS ID, they can request information about where the underlying files are stored using that one consistent URL. And it may be that that file is actually mirrored across many different clouds and regions. So they can use that DIRS URL to resolve that into a particular backend storage that may be particularly useful for them. Maybe they're working in Azure, for example. So they can resolve that ID into the closest URL, and they can use that to submit workflows in a kind of cloud agnostic way. So overall, the WES spec, in addition to TESS, DIRS, and others from the GA4GH, define a set of standards that specify how workflows should be executed, how inputs and tasks can be defined, and how to get information about workflow runs and outputs. So the idea is, again, to expose a relatively small API surface so that users are able to more easily move between environments, and they're able to use the same commands, queries, and tools they use in one environment in all of the other ones as well, without needing to have a deep understanding of the complexities of a particular backend or computational environment. So this allows for much simplified collaboration and sharing of data and processes when researchers and communities are able to align with that standard. And here I just have a few examples of workflow languages that implement either WES or TESS specs. So CWL and WDL are both WES compliant workflow languages. And Nextflow is working on becoming TESS compliant. I believe it's currently in beta. So again, I personally use WDL, or workflow description language. And just after this section, actually, I'll take you through writing a simple workflow and workflow description language and show you what it looks like. All right, so here we are. So workflow description language, or WDL, as I had mentioned, is a language that was developed by the Broad Institute. So they're the creators of GTK and other tools. I'm sure you've heard of them. It is a workflow language, not a framework. So what I mean by that is that it only defines the syntax of how the workflows are written. So the series of tasks, inputs, and outputs required to run the analysis, but it does not require a specific runtime environment. So what that means is that anyone is able to create their own implementation of a program that is able to parse and run WDL workflows, which makes it very flexible. And this is kind of analogous to, for example, the Java programming language. So Java is a programming language developed by Oracle, and Oracle, for the most part, defines the paradigms and the functions that can be used to write Java programs. But since Java is just a standardized programming language, it's a syntax. When you're running a Java program, you don't actually need to use the runtime environment produced by Oracle. So what that means is that there are open source versions of the Java runtime environment which implement the same protocols and understand the language in the same way as the runtime environment produced by Oracle. They just implement those protocols maybe differently, but as long as they're producing the same outputs and they understand the same interfaces that are exposed by that syntax, then you're able to use kind of any execution engine that you want to. So WDL is kind of similar to that, in that it's just a syntax and anyone can write their own execution engine as long as it's able to understand that programming language syntax. So this has huge benefits because it means that if there's something you're kind of unhappy with in the implementation of a particular runtime environment, or you think there's some inefficiency, you don't like how a particular part of it works, you can write your own, or you can even extend the current ones to fix that inefficiency. And so again, this means that workflow description language it's not opinionated about how the workflow is executed. It's really just a kind of recipe for the series of tasks that need to be run and how to channel inputs and outputs from one task to another. So because WDL is not coupled with a particular runtime environment, a number of different implementations have been built that support different backends. So for example, there's Cromwell, MiniWDL, and DXWDL, amongst others. The big ones are definitely Cromwell and MiniWDL. So Cromwell is what Terra uses in its backend, and Cromwell supports local backends, HPCs, and several of the cloud providers at varying levels of support. So Google, Azure, and AWS. There's very tight integration, particularly between Cromwell and Google, but it's moving to the other clouds as well. There's also mini-WDL, which works very well on AWS and locally, and hopefully someday it will move to the other clouds as well. But this kind of just gives you an idea of some of the flexibility in that these are all open source projects that are built by the community. And if you find that you're using, for example, mini-WDL with AWS and you want to move to GCP, you're not out of luck. You can just use another execution engine. And again, because WDL syntax, it's just a language or a syntax that is understood by these different engines in the same way. You know that it's going to execute the same in any of those different engines, as long as they work on that particular backend that you're looking at. So the idea with WDL is that, quotation marks, it runs the same on every backend. I have a little footnote there. That's the stated goal. But of course, there's always idiosyncrasies, but that is the goal of it. And because it is a standardized syntax and language that can be understood by any of these execution engines, that means it's very easy to share, it's infrastructure independent, and it's easy to make it reproducible. So what actually is workflow description language? Well, it's a syntax and a programming language, and it basically, what it does is it wraps arbitrary shell command lines, and it lets you define the inputs and outputs for a particular command that you want to run or set of commands, and it lets you funnel different outputs of tasks from one sequence to another. So it lets you basically make your analysis and chain these tasks from one to the next. And basically any tool that can be called from the command line can be written into a WDL workflow. The user is able to specify the resource requirements, so threads, RAM, disk size, and the runtime environment, so the container that the tasks are being run in for each task. And there's two main keywords in a workflow description language workflow. One of them is the task. So this is the kind of logical unit of work, and you can break this up any way that you want to. You're not restricted to running a single command. You can run several in a row, or you can run a script that calls many different programs. But the way that I usually try to break my tasks up is looking at logical units of work. So for example, aligning reads to a reference or sorting a BAM, I would make those separate tasks because they have separate goals. The task also defines the command itself, or the set of commands that you want to run. It defines the inputs and the outputs of that task, and the environment in which the command should be run. So as I was saying, the CPU, memory, disk size, and container. The other part of the WDL workflow is the workflow itself. And what that does is it calls tasks in an order. And so it just determines in which order they should be called and run. It lets you do fancy things like scattering. So you can, for example, if you have a file that is sharded by chromosome, for example, you can run the same task on each of those different chromosome shards. It defines how inputs and outputs are linked between tasks. So if you run task A and the output of task A is the input to task B, it lets you define that. And it tells you also the inputs and outputs of the workflow at large. So there can be many, many, many tasks in a workflow, but there's only one workflow definition that is calling those tasks in order, basically. So now I'll take you through a pretty simple conversion of a particular command line to a WDL workflow. Excuse me, sorry. So in this instance, we might want to create a workflow that's going to calculate statistics on a VCF file. So the first step would be indexing the VCF file. So you have your VCF file, you end up with a VCF index. Pretty simple. You could use something like tabix to do that. And in our second step or task, we will calculate statistics on that. So we could use something like BCF tool stats. So breaking this down into inputs and outputs of each task, the input to task 1 is, of course, the VCF file. The output is the VCF index. The input to task 2 is both the VCF file and the index, and the output might be the statistics file that we're trying to compute. So if we were running this interactively on the command line, this is what it would look like. We just run tabix, very simple. And for task 2, we run BCF tool stats. We redirect the output into some sort of file. So if we're writing this into a WDL, here's what it looks like. First, we have the task keyword followed by the name of the task. So you name it something that makes sense to you. So in this case, I've called it index VCF. We surround the whole task with these curly braces. And then you have the command block. So this is specifying what is actually going to be run. And you can see that this is just exactly what I was running interactively on the command line. So I'm tabixing that VCF file. You also have to define the input. So here's where we can take this and make it more generalizable. So you're not just hard coding a particular value for this NA12878 VCF. You can do it for any VCF. So we can replace the name of that NA128 VCF with this variable that we have defined in the input section. So in WDL, inputs and outputs are typed. So in this case, we know that this is a file, and that just kind of tells the backend how to interface with that particular input. So if it's a string input, it's not going to do anything to it. It'll just pass it through. Whereas if it's a file, if you are in a cloud environment, for example, what's going to happen is we're going to spin up a virtual machine to run this task. And it knows if it's a file input, it's going to have to copy that file into the virtual machine prior to running these commands. So all of these different typed inputs are handled a little bit differently. Next, we have the output of the task. So here, again, it's just the index, and we can also, once again, use variable syntax here. So we know that the index file is going to be named and located at the same path as that input VCF file. It's just going to be appended with .tbi. So that's what task 1 looks like. It's pretty simple. And there's one kind of section that I'm missing here, but we'll come back to it at the end, and that's the runtime block, which is where you're able to specify CPU and memory and things like that. So again, just looking at our simple flowchart, task 1 inputs VCF file, then you run your command, then you have your output. Very simple. So I'll whiz a little bit faster through task 2. So again, we have the command, we run BCF tool stats, we redirect into a file. Same as the command line we run interactively. Here we have 2 inputs. They're both files in this case. So we can replace this previously hard-coded name with the VCF file variable. You can see that we're still redirecting into something called NA12878, but there are also functions built into WDL that would let you kind of abstract that away. For example, there's a base name function. And also, because you're running all of these commands just in a normal shell, you can also use any utilities that the shell has available to you. So you could just use the base name function that's built into Bash, for example. So you end up with your output file. We call it by some name. We specify the location to it, and that's that. So you got your inputs, your command, and your outputs again. So very simple. So now I'll move on to the environment part of this. So WDL workflows are able to manage their environment using containers. And the final step in ensuring that a workflow is running the same in every environment, so regardless of the backend, cloud, local, HPC, it's going to run the same everywhere. And the way that you ensure that is using containers. So as I mentioned, it uses containers to manage that environment. And a container defined by Docker is a standard unit of software that packages up code and all its dependencies so that an application runs quickly and reliably from one computing environment to another. You might have heard of some of these container services. There's Docker, which is the big one. I think it's quite well known. And there's also Singularity. Singularity is used, in my experience, more in academic realms. And the reason for that is that Docker requires some pretty intense root level permissions that a lot of sysadmins are not really willing to give, whereas Singularity is able to run without root level permissions. So thankfully, they're pretty interoperable. You can use a Docker definition to make a Singularity container. So that's another standards win. So what a container does is it wraps up tools and all their dependencies into a sort of self-contained unit. So they're similar to a virtual machine, but they have less overhead, if you're familiar with virtual machines. So it's kind of like a small isolated computer that consists of, at the base level, an operating system. So this is usually, but not always, a Linux distribution. So for example, Ubuntu, Debian, CentOS. There are also OS X, I don't know what that's called, Mac OS and Windows virtual machines, but they're a little bit more complex. Particularly, I believe Windows virtual machines and Docker-based images require licenses. So they're a little bit more difficult to use than Linux-based ones. And most bioinformatics tools use Linux anyway. So you have that base operating system image, and then you can also install any libraries and dependencies that you need for your set of command line tools that you want to use for your particular task. So that could be Python modules, it could be R libraries, it might be C files or operating system libraries. And finally, you install any required command line tools. So for example, I might install SAM tools or JTK, anything that I need for my particular task to execute. Docker Hub is a website that you might have heard of, and it acts as a repository for a bunch of pre-built containers, so you can pull and use these freely. It hosts a really big selection of these base layer operating systems, as well as containers that are more complex and that have for example, specific versions of programs installed into them. So, for example, SAMtools is on there. Some of them are officially distributed by the maintainers of the operating system or the programming language in question. So, for example, the creators of Python maintain different Docker images that have different versions of Python on them. But for bioinformatics tools in particular, using public Docker images that you can find on Docker Hub can be really useful because it means you don't really have to worry about installing the programs on your computer itself and potentially messing up your environment with installing all these possibly incompatible dependencies. Instead, what you can do is you can pull a Docker container that already has a particular application installed, and you can just try it out and poke around without actually affecting your host system, which is really, really nice. So this is what Docker Hub looks like. And here I'm showing you the official repository for Ubuntu. So you can see it's tagged as one of the Docker official images here, which means that the maintainers of the Ubuntu operating system made this and they distribute it for use. So these like base level operating systems like Ubuntu are really handy to use as the bottom layer of your Docker image that you want to build, where you can kind of build up a minimal operating system image and add only those tools and dependencies that you need for a particular tool that you're interested in. And this is what you see when you click on, you can see in this previous image, there's this like tags tab. So when you click on that tags tab, this is what you see. So there's a whole bunch of different versions of Ubuntu. They're named kind of wacky, but that's just Ubuntu's paradigm. So there's Xennial, Rolling, Groovy, et cetera, et cetera. So you can see that not only can you get the most up-to-date version of a particular operating system or tool, they also typically will have a kind of back archive of older versions in case you need a specific version. Here's an example of Broad Institute's Genome Analysis Toolkit. So they will package up some underlying operating system, install everything they need, like Java, and then they'll add GATK to that image so you can use it. And again, they have this really handy back archive of different older versions. So if you need like 3.6 or something like that, they will very likely have an image there that you can just pull. So now we'll go into an example of how a Docker container is defined and built to kind of better understand them, because I know that containers and images are kind of an abstract concept. So I've, up until this point, been using two words to describe these Docker objects, images and containers. So an image is the object that you pull from Docker Hub, and it defines the different layers, the operating system, the packages and tools that are installed. And when you run an image, for example, interactively to use the tools that are packaged inside, the running version of an image is called a container. So again, the image is the definition of the tools and packages that are installed, and that is built. And when you're running a version of that, it's a container. So the way that you define the different layers of your image is by using a Docker file. So a Docker file is defining each of these layers. So anything that you see here highlighted that starts with capitals, that's a command to Docker, the program, and Docker knows how to interpret that. So when you say from, what you're saying is make the base layer of my image whatever follows. So this is just an image tag. In this case, it's Ubuntu version Xennial. And then all of these other commands here, run is a really common one. Again, those are commands to Docker, and Docker will understand what to do with that. But every time you see a capitalized command starting a line, that means it's going to build another layer onto that image. So here we have 4 layers because we have 4 of these capitalized commands. So the base layer is Ubuntu Xennial. And then maybe we want to install SAMtools, for example. So we would use the run command. And what that does is within that base layer of Ubuntu Xennial, with all of the programs and interfaces that would be available to you if you just were running that operating system locally, you run this command. So it's as if you had an interactive terminal in there. So it's going to run apt-get-y install SAMtools. So what that's going to do is, of course, install SAMtools using the apt repositories. And it's going to, when you build this image, it's going to run that on your machine. And if it succeeds, then you'll end up with an image that has Ubuntu Xennial and also SAMtools installed on it. So it will have 2 layers in it now. And then with progressive layers, you can add other things. So for example, here we're installing Python 3, and here we're installing NumPy using pip. So similarly to how a WDL file, you're just wrapping a command line, and anything that you could run in that environment, you can put into that command. It's the same deal with when you're building a Docker image. Anything that you could run in that operating system with the tools you have installed in the previous layers, you have access to in these run commands. There's also a few other commands. So for example, there's copy, add, environment. Those are all also useful, but run is by far the most common one that's used. So as I was saying, everything after the run command is a command that's running on that built Docker image. So again, layer 1 is the base image. On top of that operating system, that base image sits SAM tools. We install Python, and then we install perhaps a module using pip. And you can go on and on and on until you have as many.
Live streaming is on.
Okay, here I go. All right, so looking at this kind of diagram from the ground up, we have our base image, Ubuntu Xennial. We install all of these layers on top of it until you get to the top layer. So as an example, if I wanted to run a task that was, say, BWA-mem, I would maybe I want to install it from source. So I would run a task to install Git. I would run a run command to git clone that repo. And then I would have a command that would cd into that repo and run some make command or something. I can't remember how BWA is installed, but the idea is anything that you would do to install a program locally, you can do when you're building a Docker image. And I think one of the most important and key points about Docker images and containers is that the image itself is read-only. So you build this image and you kind of finalize it, you tag it, and you push it so it's available to others. And then when you're running a container based on an image, that's a sort of transient top layer. So when you're in an image and you're running it interactively, you have access to all of these tools that you've installed. You can copy files into and out of that Docker container. You can mount volumes and things like that. But any changes that you make to the operating system, for example, if you install additional tools, if you edit a config file, when you shut down that container, those changes are totally gone. So all you get really is this kind of like transient kind of environment with all of the tools that you have installed. And what you want to do is you pull in your files that you want to analyze or process, you run that processing, and then you have to make sure that you copy those out before you shut down that running container, because any changes that you make are not going to persist. And that's a nice thing, because it also means that you're able to kind of work in that space freely, and you know that you can't actually mess things up too badly, because all you have to do is shut down that container and start up a new one, and you'll get the same base image that you had the previous time. So going back to WDL, now we'll slot in where Docker goes. So we have the input, the command, and the output, and now we have this final runtime block. So you build your Docker image, you put in all of the tools and dependencies that you need, and you know that if people are pulling and running this workflow, they're always going to be using the same versions of the same tools. And this is particularly useful when, for example, I've worked with a lot of collaborators that want to either reproduce the results of a very old paper that might use tools that were published a decade ago, or maybe they just want to make sure that their current analysis is going to be reproducible in the future. So if you make a Docker container that has all of those tools, even if they're not maybe available in the future, you know that you have saved this environment that is going to be able to run. And even though maybe the repos underlying some of those tools might not exist in the future, as long as you have that built Docker image, people are still going to be able to run your analysis kind of throughout time. So the runtime block is also where you define, as I mentioned, some of these other parameters that your virtual machine might need. So you can define the number of cores that are needed, the amount of memory, how much disk space might be needed. And there's a bunch of other runtime attributes that can be defined here, but they're less important, and some of them are more specific to different backends than others. So this is really handy, because what it means is that you can determine exactly how much resources a particular task needs. For example, maybe I know that BWA-mem will fail unless it has, say, 16 gigs of RAM or something like that. So I can specify that that is the minimum amount of memory here. And when this WDL workflow is spun up by any of these different backend executors, it's going to make sure that it provisions me a VM that is at least 16 gigs of RAM. So it's less likely to fail going forward. All right, so conclusions. Adhere to community-specific specifications to ensure interoperability wherever you can. Rather than branching the spec, try to get involved if the existing standards don't meet your needs. Contribute to open source projects wherever possible. Share data early and often. Open source software and science is just better for everyone. It allows for more rapid iteration and scientific discovery. And again, some of those discoveries can be life-changing and life-saving. So it's really important. It also increases statistical power and enables community validation of your methods and analyses and your results. And finally, containerize tools and dependencies in order to ensure sharing and consistent results. This allows other researchers to validate what you're doing, and it also allows you to potentially harmonize and co-analyze varied data sets through space and time because you have this consistent workflow that you know is always going to run the same. So thank you very much for listening to my presentation. That is all. Does anyone have any questions?
Thank you so much, Heather, for your presentation. It was very great to learn more about what I knew before, and it was very well presented. I don't know if you have a question. You can ask questions, Sandrine, if you want.
Oh, sure. I mean, I just wrote it in the chat, but I really enjoyed your presentation of WL in particular, because this was something that was new to me. So thank you. But I was wondering, do you know if there's any downsides to using that as opposed to any other way of building a workflow?
Sure. One downside that I think exists is that it's kind of, so since it's a community-led project, it's something that's made by individual contributors, and it's not led by a company. It was initially made by the Broad Institute, but then donated to the community. What that means is that the development can run slower than one might hope in many cases. So there is active development in the WDL community, and the specification is being updated constantly, and the backend executors are also being updated in line. But you're kind of dependent on the timeline of these individual contributors who, for none of them, I don't think it's their full-time job. It's just a kind of hobby project. Or in the case of DNAstack, one of our technical leads, Patrick McGee, is on the WDL governance committee. So for him, it kind of is a job because we use WDL here. But that is definitely a drawback, is that you kind of have to wait for these big improvements until the spec is ready. But that being said, because there is this openness and community contribution, it also means that kind of anyone can just like post an issue and say like, hey, I think the spec would be made better because of this, and people will just implement that. So it's really cool.
Okay, great, thanks.
I don't know if you have more questions, but I have one. So you said on your task one for input file, VCF file, and you said that we need to put that VCF file. I'm wondering if you do the description of VCF file that this type of regex or pattern of files that it has to be taken on the workflow.
Okay, let me go back to that. So, oops, sorry, there's a lot of slides in here.
No worries.
Okay, so this one, are you asking if I input something that's not a VCF file, what will happen?
No, how do you know that, how the WDL knows that the VCF file is VCF file? Did you do certain things in backend?
In this case, the WDL does not know anything about whether it's a VCF file or not. I've only named this variable VCF file for readability's sake, but I could name that anything I wanted to. I could name it like stats or something like that. All that WDL cares about is that it is type file, and then you're filling this variable VCF file with something. And I didn't really give any, I guess I didn't have any slides with the workflow itself, but when you're running the workflow, you'll have an inputs JSON file. So you'll have an input called indexvcf.vcf file, for example, and you will take that key and you'll fill the value with the path to the actual file itself. So it could be like a cloud path, like an S3, or a local path. So you're filling out that variable with whatever you want to. And if you gave it not a VCF file, it would, it would be confused here and it would probably error.
Okay, see, because I thought that it was more about the regex of the file, for example, all .vcf that it takes.
Right, yeah. Yeah, and there is regex available in WDL. There's like a series of functions that you can use to kind of parse strings and things like that. But in this case, we're just looking at the name of a variable, and we can use that variable within the task in different places, but you have to fill in what the input file actually is.
Okay, I see. Perfect. I'm wondering also what task execution service do. You name it in your presentation. I was wondering.
Yes. I am really not the best person to ask for that, but it's largely involved with, well, it defines the inputs, outputs, the command, and I believe the runtime attributes of a task, and where to find the log, so the standard in, standard out. And you can roughly map a test task onto a WDL task. They're roughly one-to-one, but it's honestly something that's pretty abstracted from my day-to-day. Like I write WDL workflows, and I just trust that test is happening in the background. So I'm sorry, that's not a very good answer.
No worry. I was more, when you said test, in my mind is you test a function of an algorithm. So it was very confusing for me when it's on the workflow. That's why I was confusing.
Understood, yeah.
Great. And you say on your presentation that one of your job is to ensure that your workflow is accurate. So I'm just wondering, how did you know that your workflow is still accurate day by day?
Yeah, that's a really good question. So when we're first writing the workflows, we will seek out gold standard kind of truth sets. So for example, NA12878 is a really good one to run variant calling on, because there's like really good outputs from that we can compare our outputs to if we're doing something like variant calling, for example. So we'll look for a concordance with known gold standard sets like that. And we will kind of confirm that everything looks right before we run a whole bunch of samples through. And in addition to that, we're currently working on a kind of more granular testing framework where basically once we've confirmed and validated that the workflow looks good by comparing the outputs to sort of validated standardized outputs, what we'll do is we'll run the workflow once on a particular input, and we'll get all of the intermediate files from every task. And we have this testing framework now that when we publish a commit to GitHub that changes the workflow, it's able to detect any tasks that have changed, and it will run just those tasks and compare the output generated by that run of the task to the output generated by our initial run. So that means that we're able to change tasks in ways that are not breaking to the workflow, and compare those outputs and validate that they're still producing the same file or close enough to the same file. Sometimes the tools have stochasticity, but largely the same file.
Nice. I have a last question, and there is a question on YouTube live. So how did you get an error or an issue in reproducibility when you use Docker?
Sorry, can you repeat the question?
Did you get some reproducibility issue in your result when you use Docker?
I have not experienced that. As long as we're using the same image, then it has seemed to produce consistent results. Again, barring some tools do genuinely have stochasticity involved in them, like you can sometimes set the random seed, and in that case you'll have much more close results. But so sorry, excuse me.
No worry.
Yeah, as long as we've been using the same Docker image, then we have not really seen differences in outputs.
Nice. I have a question on YouTube from Sandrine Morera. So she said, does the WDL additional layer make it more challenging to do debugging?
I would say not, and that's because most of my debugging and development of the workflows themselves, I do interactively on the command line. So I'll write the Docker image, I'll start up a container, and I will run some little test sample as I'm developing the workflow, such that I know that everything looks right, and I don't have some syntax error, for example. And then once I write them into the workflows, one of the benefits of the specification and of WSPEC, for example, is that it captures the standard error and standard out of every task. So you're not losing those logs ever. You don't even have to, anywhere in here you can see I'm not specifying to keep the standard error and standard out. It just automatically is kept. So if there's ever an issue, I'm able to go in and look look at those files and try and figure out what's gone wrong. If it was a syntax error, maybe there was a transient timeout or something like that. And in addition to that, there's different configuration options, obviously for different execution engines. But for example, for Cromwell, which is one of the execution engines that can execute WDL workflows, you can set one of the flags to keep intermediate files. So usually when a workflow completes, any files that are just the outputs of tasks but not the outputs of the total workflow are deleted to save space. But you can set that to not be the case. And that means that if your workflow fails partway through, you can kind of look at the files up to the point where it failed and try and run the next step to see what probably went wrong. So there's a lot of information available. I find it pretty easy to debug.
Nice, thank you. Yes, I think the last question from Christina, she said, thank you for your presentation. What do you enjoy most about your job, and what is the most challenging aspect of your job?
I think the thing I enjoy the most is definitely the fact that it's I have constantly new projects to work on and new kind of areas of disease and new data types that I'm always looking at. It's one of the reasons why I went into bioinformatics following my master's, is because I looked really, really deeply into something for 2 years, and I found that like I was kind of dying a little bit. I just really, really got bored. And so having constantly new projects is fantastic for me. I really, really like finding bugs too. It's very, very satisfying. And as for the challenging aspect, I would say that it's, for me personally, it's learning how to become a leader without having had myself much experience being led. And that's not to say that leaders in my company are bad. It's just I was alone on a team for like 4 years, and I kind of just reported to myself. And like every once in a while, my CEO would check on me and be like, "You good?" And I'd be like, "Yep." And he'd be like, "Okay." So now that I have a team that's under me, it's me trying to figure out how to navigate that and how to be a good leader. So I'm like reading a lot of books. I don't know.
I know what you mean. It's a big challenge. Perfect. I think we are done for the questions. Thank you so much for your presentation again. And thank you for being here for this presentation of this month. And see you next month.
Awesome.
Thank you so
much.
Bye.
Bye now.